So before we did monohybrid cross, now we are going to do dihybrid cross. Now in dihybrid cross, it is a cross between two parents differing in two pair of contrasting characters. Mendel made some dihybrid cross in pea plant. Example, cross between pea plant with round shaped yellow colored seed and cross between wrinkled shape and green colored seed. Now we will see the crossing of dihybrid cross. Now here we have taken two parents. The first parent is round yellow. The phenotype is round yellow, while the genotype is capital R, capital Y, capital Y. This is the dominant character. Wrinkled green is the phenotype of the second parent, while the genotype is small r, small y. This is the recessive character of the parent. Now, we take the gametes, that is capital R, capital Y, small r, small y. Now, we pollinate this, and the phylyl 1 generation, that is the F1 generation we get, is capital R, capital Y, small y, which is a round yellow. Now we do the selfing of this round yellow plant, and we get the gametes as capital R, capital Y, capital R, small y, capital R, small y. Now crossing will take place between these and these gametes to produce the F2 generation, which is the phylyl 2 generation. Then using the Punnett square, we get the information of F2 generation gametes produced. The dihybrid phenotypic ratio of the Punnett square we get as round yellow 9, round green 3, wrinkled yellow 3, wrinkled green 1. Therefore, the phenotypic ratio is 9:3:3:1. Now, the dihybrid genotypic ratio. Genotypic ratio is this we get using the genotype capital R, capital Y, and so on. We get as 1:2:1:2:4:2:1:2:1. Now, before we have done the two laws of Mendel, which is law of dominance and law of segregation. Now we will do the third law, which is law of independent assortment. What does this law says? Let's see. When more than one pair of characters are involved in a cross, factors, pairs or gene pairs independently segregate from each other. So this means when one or more characters are involved, then the genes are separated independently. For example, we have done this dihybrid cross from which we get so many gametes. So this is the independent segregation of genes from each other.